Good evening. My name is Brooke Ochterman, and tonight I am going to be speaking over the topic of superenhancement and control of AMH expression. This paper was written by David W. Dresser, and the purpose of his paper was to identify if mutations in the Wilms tumor factor or mutations in the downstream enhancer sequence affect the superenhancement or expression of AMH. In his experiments, mice Sertoli cells were used because they are easy to compare with humans. And I picked this paper because I thought an AMH is also part of sexual development. And I chose this paper because I just thought sexual development and differentiation was based on the X and Y sex chromosomes, because that's what we're taught in school. And upon further investigation, I realized that there are other components that help differentiate male and female. So in order to understand the paper, there's some background information that needs to be acknowledged. And for one of them is to understand what AMH is. So AMH, or anti-Murine hormone, is a glycoprotein of the transforming growth factor beta superfamily, and it plays a role in forming male and female reproductive organs. It's found in the Sertoli cells in the testes of men and in the granulosa cells in the ovaries of females. If males have an underexpression of AMH, they could get persistent Murine duct syndrome, which is indicative of both sexual or reproductive organs in the male. And women who suffer of overexpression have polycystic ovarian syndrome, which causes prolonged and irregular menstrual cycles, and it affects their egg count and their follicle production, and it also affects their fertility. The figure here illustrates the 3D quaternary structure of AMH as shown through the alpha helices and the beta sheets. What is Wilms tumor factor element, also known as WT? It's an activator for AMH receptor gene, and biochemically speaking, it's a transcription factor with 4 zinc fingers located at its C terminus right here, and it plays a role in urogenital development. And if it's expressed, the Wilms tumor is expressed, which is a form of pediatric kidney cancer that affects 1 in 10,000 children. So it's a very rare form of cancer. And then what is a downstream enhancer sequence, denoted as DE? It is a enhancer for regulatory DNA sequences that bind to specific proteins and enhance the transcription of that gene. So for example, the downstream enhancer sequence here enhances the AMH gene in the transcription of that gene. It can be either downstream or upstream. However, in this example and in this paper, it is downstream. And since we used mice Sertoli cells to compare to humans, we had to divide the DE into 3 subregions. And then what is the WT-DE2 bridge? It is illustrated here through the dotted line, and it is a connection of the enhancer here to the promoter through a specific duplex factor. This indicates that the WT located between the tata box and the start of translation for this gene and the downstream enhancer sequence 2 are anchor points for this duplex factor, and it brings remote DNA up to the promoter for transcription. So suppression versus expression of AMH. In order to have an expression of AMH, you need to have an XY chromosome, and the SRY is the sex-determining region of the Y chromosome. So if you have the SRY, there's a network of hormones and connections that cause the gonads to turn into a testes. And then the testes create the Sertoli cells and the Leydig cells. The Leydig cells create testosterone, which create the Wolffian duct development, and that leads to the formation of the male reproductive tract. The Sertoli cells lead to the MIS, which is another word for AMH, and that causes the Murine ducts to degenerate. However, if you are female, you do not have a Y chromosome, therefore you do not have the SRY, so the gonad converts into an ovary, and there's no Sertoli cells, there's no Leydig cells, there's no testosterone, there's no AMH, the Wolffian ducts degenerate, therefore the Murine ducts develop, creating the female reproductive tract. The focus of this paper that I'm focusing on is the ablation versus the expression of enhancement of AMH. It was illustrated through the paper that mutations at site 1 in the DE, located right here, this is site 1, and mutations in the WT, located here, created ablation of enhancement and expression. And then this also verified that the WT and the DE2 are anchor points for the specific duplex factor, bridging remote DNA to the promoter for transcription. At site 4, located between DE1 and DE2, there was a level of AMH enhancement, not ablation. How did they understand that there was expression? They used a method called flow cytometry, and flow cytometry measures the size and complexity of single cells. The fluorescent dyes, the eGFP, was bound to the DNA of AMH, and it emits at a higher wavelength because the fluorescent dyes excite to a higher energy state. So the single cell suspension here flows through the sheath of isotomic fluid, creating a laminar flow, or one at a time, as the cells drive down into this interrogation point. After they go through the interrogation point, they connect with the argon ion laser, and the light scatter is emitted and collected through optics and dichroic mirrors, which are then filtered into electronic amplification and converted for computer analysis. The result of this experiment of the Sertoli cells was that the emissions were red and green, therefore autofluorescence could be excluded. Another method used in this paper was the double overlap PCR method. In order to understand the double overlap, we must first understand PCR. PCR, or polymerase chain reaction, amplifies small segments of DNA and builds new strands using the originals as templates. So you have the original, and then you build new strands. So one's original and one's a new strand. One's original, one's a new strand. And then the double overlap involves mutations that are inserted at specific points of the developed DNA strands, and then the PCR method combines them, creating a large strand and a large supply of mutated DNA in order to be analyzed. So the result of the experiments are that the mutation at site 4 in the minor loop of the downstream enhancer sequence results in super enhancement of AMH, which is shown through eGFP expression. So if you look at A, the graph for A, the control has very low expression of eGFP, but the mutations of site 4A and 4B illustrate a larger enhancement, which is like almost tenfold. And then the mutation of the WT, or the Wilms tumor factor, illustrates an ablation of enhancement as shown in B. So the control for B is a lot larger than the mutated WT and DE site 4. This could be indicative of a disruption between that WT and the DE2 bridge. Therefore, the conclusion of all these experiments is that you need a downstream enhancer sequence for ablation of AMH expression. For future studies, it would be interesting to see how WT mutations at various other genes can affect cancer immunotherapy, and how also the AMH expression or enhancement or ablation can help women suffering from infertility and help those men that suffer from the Mullerian duct syndrome. And these are my references. Thank you.